What makes our minds tick? That's exactly what my lab at Johns Hopkins studies through what we call molecular photography of the protein machines that enable our brains to think. What I'm excited to share with you today is a little bit about what One Mind is funding, where recently we've unlocked a brand new target for developing new therapies for targeting diseases like schizophrenia and anxiety, called GluD1. But before I get started, I would like to give you some insights into my lived experience and how that shaped my research. So this was back in 2019 when I was a postdoctoral fellow at Harvard Medical School, and I was studying how our cells recycle proteins. And I think we can all agree this looks like a decently beautiful mind up here with the MRI scan. But the other half does not. And so this is the result of a snowboarding accident where I had a subdural hematoma. And thankfully now I'm fully recovered from this, thanks to a craniotomy. And while it began a battle with some severe anxiety, I was quite thankful for it because it really helped me reshape my scientific approach back to the protein machines that really make our brains tick. How I like to think about our brains is that they're really universes of synapses. So I think and what I mean by that is I think perhaps, after a couple glasses of wine later when we're all staring up at the sky, something we can all appreciate is the true vastness of the universe. So here I have an image from the James Webb Space Telescope where it's projected there's about 2 trillion galaxies in the universe. But our brains are actually much more vast and complex. So here I'm showing an image of synapses. So what synapses are these junctions between the cellular foundations of our brains, which are called neurons. And it's projected that we have upwards of 100 trillion of these in a human brain. So what my lab is quite interested in is understanding the molecular machines that are going to enable communication between cells at these junctions and how that might go awry, because a growing appreciation that we have in the field is that dysregulation of these synapses is central to the development of psychiatric disorders. So to show you a little schematic of how these look, generally we're going to have one cell passing a neurotransmitter, which is a chemical, to another cell. So at most of these synapses in our brain, this chemical is going to be glutamate. After me, you're going to hear about, from Dr. Nir Eshel, other neurotransmitters that you may be familiar with, such as dopamine and acetylcholine. But in our case, we're interested in these glutamate receptors, and what exactly these are doing is they are going to be binding or interacting with this chemical neurotransmitter. In the center of this protein, it's going to open up this channel that allows ions like sodium and calcium into our neurons. And this generates an electrical signal that's really fundamental to the functions of our brains. And how we like to study this is with a technology called cryo-electron microscopy, which is really molecular photography. So every single one of these black dots in this image is actually a purified glutamate receptor. So I have some circled in yellow here, and they're about 18 nanometers in diameter. And to put that in a scale we should all appreciate after this weekend is you can stack nearly 17 million of these on top of each other in a single bottle of wine. So we collect nearly on that scale of images, and what we do is then we actually average them together in 2 dimensions. So what you can actually begin to see is what looks like almost an X-ray of these little receptors. And then after collecting even more of these images, we can then begin to see how these look in 3 dimensions. So when we're looking at these in 3 dimensions, we can really begin to appreciate the architecture of these receptors, which are going to enable us to understand function. So we can directly see how pharmacological targets may be interacting with our glutamate receptors. We can see if a patient has a certain mutation, where exactly it's happening in the receptor. And importantly, when we understand the architecture, we can see where we want to intervene for drugs. So just to give you an idea of how this works, what we published earlier this year in Nature is that glutamate receptors work a lot like Pac-Man, where they're going to clamp down on the neurotransmitter. And as this happens across the scale of the channel, it's pulling open that channel so then the ions like sodium and calcium are going to be able to get into your brain or to your synapses. And what's been really critical is what we'll publish next week in Nature is we've used this approach to really identify the function of these proteins that are central to the development and maintaining our synapses. There are many mutations in these GluD receptors that are directly implicated in conditions such as schizophrenia and anxiety. And for decades, we thought these have just laid dormant in our brains. But really, these receptors are very much alive. And now with funding from One Mind over the next 3 years, what we're going to be able to do is understand how these receptors function, how channelopathy mutations in diseases such as schizophrenia are altering the structure and function of these receptors. And ultimately, we're going to be able to leverage our findings to truly create new revolutionary approaches to our therapies targeting these GluD receptors for patients. Thank you.